First of all, everybody, I will switch off my camera because I do have some connection problems. Sorry for that. All right. Well, I guess we can get started now. So again, good morning, good afternoon, or good evening, depending on in which time zone you actually are. And also, I think I'm still allowed to wish you a belated, but still a very happy new year. So my name is Mathieu Vinken. I'm the chair of the in vitro and in silico toxicology speciality section called In2Tox of EUROTOX, which is the Federation of European Toxicologists and European Societies of Toxicology. So In2Tox was founded only last year, actually 2 years ago in 2019, during the EUROTOX conference, which was at that time organized in Helsinki. So by the way, if you would be interested in these and other In2Tox activities, or you would like to join In2Tox, which is possible, please go to the EUROTOX website, eurotox.com, where you can find us in the About Us section. So we usually, as you probably know, have an in-person meeting during the yearly EUROTOX conferences. However, of course, with most events these days, this was not possible because of the COVID situation. So therefore, we thought it might be nice to have like a webinar instead as a kind of an alternative. So it's probably not a surprise to you that we are actually fully focusing on COVID-19 today in this webinar, and specifically on the role of in vitro and in silico methodologies in COVID-19 research. So I need to emphasize here that we already organized the very same webinar in September, but unfortunately at that time we did experience some technical problems. So therefore we decided to repeat this webinar. So the presentations of the speakers will be the same, but of course they have been updated where relevant and necessary. So we do have two splendid speakers today. One will talk about the in vitro aspects and the other speaker will more deal with the in silico aspects. And before introducing the speakers to you, I first want to share some, let's say, household information with you. So this webinar, as you already noticed, is being recorded and will be available in a couple of days or so on the EUROTOX website. And that is of particular interest for people who could not attend today. So you will have the opportunity to actually, get back to the webinar if you want. And as always, with these kinds of online meetings these days, I kindly ask you to mute yourself in order to reduce the background noise, and also, like in my case, to switch off your camera, especially if you have connection problems. So each of the talks, as you might have seen in the program, will take about 20 minutes, and there will be a Q&A session, so question and answer, of about 15 minutes after the second talk. So I would suggest that unless you have very burning questions, that we postpone all of the questions until the very end of this webinar. So basically, we try to wrap up at about 2 p.m. So that is Central European time, of course. Now, very important is that during the webinar, I will follow up on the questions. So please, if you have questions, you can send me those in the chat box and I will take care of it and I will try to make a compilation of that. So that will be addressed at the very end of the Q&A session. So without further ado, I think I can introduce the first speaker. And again, it's my very, very great honor to introduce Professor Thomas Hartung. I don't think he actually needs introduction. I think he's quite famous, but anyhow, he's the chair of the evidence-based toxicology and professor of molecular biology, microbiology, and immunology at the renowned Johns Hopkins Bloomberg School of Public Health in Baltimore in the US. He's also a professor, by the way, of pharmacology and toxicology at the University of Constance in Germany. As you might know, he directs the Centers of Alternatives to Animal Testing, known as CAT, of both universities. From 2002 to 2008, he directed the European Center for Validation of Alternative Methods, so ECVAM, of the European Commission. And he authored more than 575 papers, which is pretty impressive, I have to say. So the title of his presentation today is New Approach Methods as Door Openers for Drugs and Vaccines, or the other way around. So Thomas, please go ahead and share your screen. And as we say these days, the screen is yours.
Thank you very much, Mathieu, for the nice introduction and having me again. As you all know, technical problems last time, which I caused, that result in no recording and in let's say, in a messed up presentation and webinar. So hello from Baltimore. You can see the sun rising in my background. and I'm very happy to share with you an updated version of where I see alternative methods at this very moment. In fact, there are door openers for the accelerated pace and some of the medical miracle we have seen. But before getting started here, I would like to make a few points. The first point is I'm a physician. I'm a physician in the leading school of public health. So when promoting alternative methods, I do so because I believe we do the best for patients and consumers. And this mask, which we actually printed, reads "saving more than animals," and I think this is really exactly what we're up to. Another point I would like to make is I'm trying to get the rubber on the ground, so I make my hands dirty. I'm involved in a couple of spin-off companies or consulting others and organizations. You see this here as a conflict of interest, I have to declare, but at the same time, I hope to take everything with a grain of salt, understanding that this is, from my point of view, implementing what we're trying to achieve. Everything I'm showing has been done by a team which is normally very social and is working at the moment like everybody else is. But there's also some good things when working now mostly from home and online. We've seen enormous uptake, for example, of our free of charge courses on Coursera. We have now since 2018, when we started these two classes, more than 11,000 active learners on toxicology for the 21st century and evidence-based toxicology. So these are classes which we offer for free. If you want a certificate from Coursera, you have to pay, I think, $49 to them. It is, if you want to hear really in-depth, each is 13 lectures of up to 90 minutes about topics relevant to the replacement of animal testing and the modernization of toxicology. So let's talk COVID. COVID is about 1 year old, and it's an incredible success story. I mean, if you think it was December 2019 where we saw the first cases, a broader audience, and the WHO was informed on the 31st of December 2019. But it was only 10 days later that the first PCR in the UK was, so outside of China, that was already described. The whole genome was revealed on the 11th of January. We had the PCR test, which became the WHO standard out of Germany on the 17th of January, and antibody tests in March. On 1st of May, the first emergency use authorization by the FDA for a drug with positive clinical data. And in December, as we all know, now 3 vaccines, if you include AstraZeneca's acceptance in the UK, were there. I don't need to tell you that this was very different, for example, when we looked at HIV, where it took us years to identify what the virus is and to come to any type of therapies. So this illustrates how the medical community has taken this up. I checked last week when I did this presentation. On the 11th of January, we already had 90,000 articles on COVID-19 and SARS-CoV-2. This is PubMed is an archive which includes about 25% of the medical literature. You have to think of all of the self-published archived materials. It's incredible. You might ask, who can read it? And actually, I believe very strongly that it will be in large part computers reading it, because we need to condense this for others. We need some systematic reviews of this literature, quality scoring, all of these things. And I think it will also impact on the way we are publishing in a more structured way, in a way that computers can actually read what we are publishing in the future. A lot is coming up here. And as you know, my involvement in evidence-based toxicology is very much along this line, but we need more preclinical science evidence-based. We started evidence-based toxicology in, you could say, 2006. Sebastian Hoffmann's PhD with me on in vitro evidence-based toxicology was actually a pre-run from 2002 to 2004. We then did the first conference in this topic. I have a chair for evidence-based toxicology since 2009 here at Hopkins, and we are proudly hosting the evidence-based toxicology collaboration, which has a newsletter and everything. If you're interested, looks for people to get involved on systematically capturing what is happening in toxicology. What we should see is that to some extent, and as the numbers of publications indicate, we see a scientific gold rush at Klondike. Everybody who can just spell COVID-19 is writing articles at the moment, me included. Most are ill-equipped because they have no background in infectious diseases, and very few get rich. And these were also at Klondike already, those who sold the shovels and sieves. In our case, these are the people who sell the reagents and the tests and whatever we're using to adapt our research to COVID-19, which creates a problem. And this problem is created by the medical challenge as fast as possible to develop drugs and vaccines, as we have seen in record time recently. And we should really understand the enormous numbers of clinical studies. Actually updated this last week. So the 30,000 studies launched and 4,400 are last week's data, not 17th of September. It is really an enormous run towards finding treatments taking place here. There's already in April last year, we had 160 vaccines under development. As you know, beside the three we know, the Russian, the Chinese, and others have come up with some vaccines out of these studies. So as we were already early on talking about what does it mean for alternative methods. And in this article, which I recommend to you, it's free of charge, available from Archives of Toxicology last year, we were discussing what alternative methods can do for repurposing existing drugs, for target discovery, drug efficacy, for vaccine development, the combination therapies necessary, drug safety, and the quality control, an aspect which is often underestimated, that batch release for vaccines is typically necessary. I believe that all of these things are very much interwoven with economical aspects. This is an article from 2018. The most important omics is economics. Again, an open access article from our journal Altex. And in this article, we described that it takes you typically 12 years to develop a drug at an investment of 2.9 billion. And you see on the lower left, the large data from Tufts demonstrating the enormous investment preclinically and clinically in producing a new drug. And this is not very different for vaccines. Vaccine development in the past took 8 to 18 years, on average 10.7 years. Interestingly, a lot of them produced in Europe and a lot of them consumed in the US, as is true for many drugs in general terms. So it was a challenge to compress a development of a vaccine into a one-year course. And vaccine production has before not been a very, very lucrative model. 10.7 years and a probability to make it to the market of 6%, with investment costs not different to those for drugs, at least not much lower. So it was not really a model where any company would invest. And we see now that suddenly it has become the goal for so many of these. And in the end, we will again only need perhaps a handful. We will see that many of them don't make it to the market because others have already taken the place. And again, this will be a few percent of the substances which are under development. Actually, this week, the new Altex is becoming available, where I'm reasoning about the whole blood monocyte activation test and pyrogen test, which I developed exactly 25 years ago, and which patent just expired. And I'm talking about others about the need for pyrogen testing for these drugs, for these vaccines especially. To give you a feel, we have for flu vaccines, for example, about 500 to 600 batches which we are releasing every year in Europe, and each of them has to be controlled for pyrogens. So it is extremely important that we, for the new tests, for the new vaccines, which also have to be pyrogen tested, can rely on alternative approaches to not bring up animal numbers again, because this group of tests has actually been one of the biggest success stories in alternative methods, because it brought an area which used as many animals as chemical testing, as many animals as pesticide testing, down by almost 80% only between 2011 and 2017 when they fully were implemented. So really, I invite you to study this example, that it is possible with a human test to really replace animal testing, if it is a meaningful test. And the practical use here for COVID-19 vaccines, for example, is obvious. So I was asking in the title, what are alternative methods doing for accelerating this and opening the door? But it seems also to be the wonder drug, the other way around, for alternative methods to really enter into our space. Let's talk about this in a little bit more detail. I believe that the important thing is how the FDA thinks about alternative methods. That's the critical thing in drug development. And the reason for this is that the US has 4.25% of the world population, but is consuming 48% of all drugs and 64% of all drugs under patent. That's the general numbers. And this is very simple why all pharmaceutical companies are mainly looking for FDA approval, and that's why the FDA is more equal than others. It's of critical importance to understand this. You have to have the FDA on board when it's about alternative methods. And it is extremely important and encouraging that the FDA has been embracing microphysiological systems. Susie Fitzpatrick is, for many of these things, an opinion leader within FDA. So I borrowed this slide from her, and it shows some of the activities of the FDA in adapting what they call microphysiological systems, the organ-on-chip type of systems. And that's another example, an impact story which was produced on evaluating the potential with these type of systems. So what we see here at the moment is that the US, who were trailing behind in alternative methods for so long, but also steadily progressing, suddenly there's they take a shortcut. They invest into technologies like microphysiological systems. They go to the airport instead of following this, and are likely to overtake in many aspects the development of alternative methods by their promotion of microphysiological systems and others. And to give you some evidence for this, it was only in December that the FDA launched a new program on innovative science and technology approaches for new drugs, which is in essence a fast track for alternative methods, which I see very much as a consequence of the experiences of last year. And there's actually a lot to talk about. These technologies are increasingly advancing. We published in Altex last year a workshop report, a stakeholder report of some 35 people who came together in Berlin a year earlier and discussed how this field is advancing towards tissue and organs on chip, human on chip type of multi-organ systems. Please read the Marx et al. 2020 article in Altex, the lower one. We had done a similar one 4 years earlier. So this is the article, which in the meantime came out, Biology-Inspired Microphysiological Systems. And you see the many co-authors here. And the expectations are really high. This is Francis Collins, the head of NIH. And he already said in 2016, when testifying to Congress, Senate actually, it was about money for NIH. I predict that 10 years from now, safety testing for newly developed drugs, as well as assessment of toxicity for environmental exposures, will mostly replace animal testing for drug toxicity. You can read the rest yourself. So this is, I think, from top of NIH, showing expectations. So if you look now into COVID-19, it is first of all a human respiratory disease. But what we learned the hard way over the course of last year was that it affects essentially all organs. Everything is affected by the infection. Rodents can do the lung. No, they can't. They cannot be infected. The animals which can be infected are macaques, hamsters, and ferrets. Typically show relatively mild infections of the lung, but we have not many reports about other organ systems being affected as they are with humans. But our microphysiological systems actually check all of these boxes. Many of these things have been studied over the course of last year in microphysiological systems for these various organs. To give you just the very first examples, instantly the commercial lung models have been studied in various articles, both the European and the US one, because that was the natural, and you find tons of articles on these things. The more complicated or complex Harvard Viss lung model, which is actually a real microphysiological system, was instantly also applied to this. And these type of systems have been installed in biosafety level facilities to make the disease studyable. We're trying to push the whole field on microphysiological systems, for example, by taking up good cell culture practices again, something I started in 1996 actually with the first Berlin workshop, and we published GCP 1.0 in 2005 before we had these type of models. And if I say these are from the last STIF meeting in Berlin, beside myself, Glenn Stacy, David Pamias, and Sandra Kucke, some of the really leading figures in getting all of this running. We published the draft for 2.0 last year. It is still time to be part of the voting now on the stakeholder changes suggested. Please register with CUT if you're interested to be part of this discussion. And we hope to release this year, finally, GCP 2.0. We're also promoting this by creating a World Summit on microphysiological systems. Please be aware if you saw earlier announcements, this has been shifted now by 6 months into actually 2022, New Orleans 2022. And we do have 2 online events leading to this in June and December of this year to be announced. And you see Susie Fitzpatrick from FDA is with us, some 35 organizations supporting this World Summit. So this will be something to stay tuned in for. Quickly, I would like to show you just with our own work and example how microphysiological systems are doing the trick in helping with these diseases. Many will have heard about our mini brain model. It is not the first, but it was the first mass-produced and standardized model of its kind. And when it became very early clear that somehow COVID-19 is affecting the brain, is leading to infections, 30 to 60% of patients show some type of neurological complications, as we learned. I very quickly asked in March, can the brain be infected? And it took a while to find the possibility to actually test this, find collaborators in virology. Our brain model had been used before for HIV, JC virus, dengue, and Zika virus. That's one of the publications. So we teamed up because we had some indications that coronaviruses can be neurotropic, so hit the brain. So we teamed up with Bill Bissai and Corinne Bullen in infectious disease here in Hopkins. Then we saw late May the first indications that here this is a neuronal cell which is infected. Every red dot is a virus particle. So this is really productively infecting human brain cells. The Financial Times and some 200 others reported about our findings. And we published this then in Altex, again, demonstrating after peer review that this is the team, my team behind it, Helena Hochberg, Fena Sille, and my wife, Lena Smirnova. And they have good reason to be worried about the children developing. So this work, which it's half a year old, this is ages in COVID-19 research, showed the presence of the AC2 receptor in all stages of the mini brain development, the infectability of some cells, and it showed it also that replication takes place and shedding of the virus by PCR and others. So you see them. There's a lot of ongoing work. Which are these cells? What is the pathology induced? What is the role of the blood-brain barrier? Does it protect others? Is it actually developing new development? And stay tuned, some of this is coming out in papers shortly. From my point of view, this has some impact. For example, on the vaccine trials, I wrote that I considered it unethical to have volunteers infected with COVID-19 if you might set a lifelong infection of the brain by infecting them. You don't know what this means for Alzheimer and others. I also added a component which I did not discuss in September because it's not only in vitro. From my point of view, there's also in silico increasingly taking up. I have an interest in this field. I'm the field chief editor for Frontiers in Artificial Intelligence. So I'm engaged in this journal. So I'm having a little bit of an overview what is happening with respect to COVID-19. And when I looked in November the first time, it was a bit disappointing. We had 75,000 scientific articles in PubMed, but only 300 were using machine learning or AI. And these 75,000 in these 2 months have been increased to 90,000, so 20% growth. But quite interestingly, we had an explosion in the field for AI, 300% growth with 1,200 papers now. And this makes perfect sense because you can apply machine learning only if you have data. And the data were only becoming available over the course of this time. Now it is the time of helping, interpreting, and mining these data. Last week, we had a meeting, a Keystone meeting sponsored by Frontiers on the promise of AI for in these pandemic times. The recording is becoming now available, so I encourage you to register if you want to see the recorded talk on this topic. So let me come to an end. The medical challenge of COVID-19 prevention and treatment to some extent illustrate the shortcomings of our animal-based drug development approach. 10, 12 years is also in large part due to the fact that it takes months and sometimes years to get the results of an animal study. Alternative methods, and it doesn't matter where they are developed, are advancing. Our key opportunities to accelerate the opportunity for drug and vaccine development and beyond, because I believe the world will not be the same also with respect to acceptance of alternative methods. So let me close. Alternative methods are enabling technologies. They allow us to do things which we could not have done with animals. And I think they are door openers for each other. Thank you very much.
Thank you so much, Thomas. As always, excellent presentation and very impressive research. So I don't see any questions popping up in the chat box. Please go ahead and do so. But again, the idea is to address them at the very end of this webinar. So then I think we can move to the second speaker of today's webinar. So the second speaker is Dr. Luca Dellafiora. He's an assistant professor at the Department of Food and Drug of the University of Parma in Italy. His research is situated in the field of food chemistry and toxicology, whereby he routinely applies hybrid in silico and in vitro methods to assess bioactivity and toxicity of xenobiotics and natural compounds. He has a particular interest in understanding and describing molecular mechanisms of action in order to extend knowledge on the effects of these xenobiotics and natural compounds on human and animal health, as well as to identify and develop strategies to mitigate the content or the adverse effects of toxicology active compounds in food. So the title of his presentation today is Studying the Interaction Between the Coronavirus Spike Protein and Human Target Receptors Using 3D in Silico Modeling. Could anthraquinone compounds prevent the interaction? So Luca, please do share your screen. And again, the screen is yours.
Okay, thank you, Matthew, for your kind introduction, and hi everyone from unexpectedly sunny Italy. So, well, Thomas is definitely right when he says that a computational method may make the difference in the research to fight COVID. And I hope in my presentation I can give you a small example of the, let's say, the enormous network of possibilities of using computational method. And in particular, the presentation you're about to see is a computational method, a structural-based method to study the COVID, and in particular, the work I'm gonna present you focuses on a specific class of natural compound, that is the anthraquinone class of compounds. So just few words to introduce myself, and that's important to understand and to read the presentation from the right perspective. I'm not a physician, I'm not a medicinal chemist, I'm not a pharmacologist, I am a molecular biologist by training, and let's say a food chemist and a food toxicologist by adoption. And indeed, my research is mainly focused on the analysis of the molecular mechanisms of action at the basis of the biological activity of food-related compounds. And that's why we started working on COVID, basically. And the reason was Emodin. In our routine analysis, we tried to map, to identify all the biological targets of food-related compound. Emodin is definitely a food-related compound, an important food-related compound, because it's let's say a natural compound, widely widespread in the plant kingdom, and but it is also produced by some fungi, and it has been recently included in the class in the number of the pseudofine emerging mycotoxins. So we can find it in depending on food in a huge abundance in specific type of food and raw materials. And among the most interesting biological activity of Emodin, there is actually the capability to prevent the interaction of the old coronavirus and human cells. I mean, old because the work I'm gonna show you was on the COVID discovered at the beginning of the 2000s. And in this very, very interesting work, Ho and collaborators show that actually Emodin was able to prevent the infection of cells by the old coronavirus in a concentration-dependent manner, preventing the interaction of the, let's say, the spike protein, and this is the spike protein of the coronavirus, and this particular region is the one highlighted in the ring here. And Emodin was able actually to prevent an interaction of the spike protein with the human cognate receptor, the human target receptor, the angiotensin 1 converting enzyme 2, AC2. But at the time was not, they didn't show any clues about the mechanism of action. So they saw that the molecule was able to prevent the interaction, but they didn't know how. And of course, if you want to develop further Emodin, or you want to derive antiviral compounds from Emodin, you must know exactly the mechanism of action. And that was exactly the aim of our work. We try to describe a likely mechanism of action for the activity reported for Emodin. The first analysis we ran was the analysis of the protein complex formation. And in particular, we used a computational approach called computational alanine scanning that aims at identifying the key residues involved in a specific protein complex. And what we saw was that actually at the spike protein AC2 interface, there were two regions, two spots of interactions that were driving basically the complex formation. Then we had a closer look at the surface, and we saw that exactly in between these two hotspots of interaction, there was a cleft, a kind of, surface groove that was potentially able to receive, to dock, to bind small molecules, and including also molecule with the anthraquinone scaffold, molecule similar to Emodin. Then we checked the theoretical capability of famidine to interact with that specific pocket. And we also calculated the capability of promazine and anthraquinone, that were the other two ligands assessed by Ho and co-worker. And we did that through docking simulations. Docking simulations mean that basically you simulate the binding event in a single frame. And as you was extremely important to see actually that the docking scores that could be proportional to the binding affinity of ligands were exactly in agreement with the data reported by Ho and co-worker, because we saw that promazine, that was the most active compound in the experimental trials, got the highest score, and amidine, that actually was in between the two, according to experimental data, got a score exactly in between promazine and anthraquinone. So this was the first, let's say, evidence supporting the possible existence of the interaction at the surface pocket in between the spike protein and the AC2 complex. Then we analyzed the interaction over the time, and we did that using molecular dynamic simulations. Molecular dynamic simulations are basically you put a complex, protein ligand complex, in a box of water, and you let it run for a specific time frame. And in that way, you can check all the ligand-induced movements of the protein. And of course, if how to say, if a ligand is able to change the organization, the stability of a specific protein, that ligand may have some specific effects on the biology of that protein. And contextually, specifically, what we saw was that actually amidine, and for instance, not anthraquinone, that was the compound with the lowest activity in experimental data, amidine was able to drastically change the organization of the spike protein. And we had a closer look of the regions that were most involved in such a reorganization. And what we saw was that one of the two regions, the two plug regions, the regions that were involved in the connection with the AC2 protein, was the one of the, let's say, region most involved in such a reorganization. In the picture in the bottom of the slide, you can see the time frame analysis of the molecular dynamic simulation. Time frame analysis means that the red frame are the red at the beginning of the simulation, the dark blue frame are the frame at the end of the simulation. So in this picture, you can see, the evolution over the time of the movement of that specific protein region. As you can see, in the case of the complex with amidine, this region, the region involved in the AC2 contact, is much more reorganized, is much more destabilized in compared to the complex with anthraquinone and promazine. So we saw that amidine, interacting with that specific surface pocket, was able to reorganize one of the two regions that are involved in the AC2 contact. Then we analyzed also the movement of the three different ligands, and we saw that amidine, but not anthraquinone, amidine and promazine, of course, but not anthraquinone, were able to persist, to stay at the surface contact of the spike protein of the coronavirus. And also in this case, we got supporting the hypothesized mechanism, because anthraquinone basically was not able to give a relevant biological effect in experimental trials, because according to the computational result, was not able to interact with the surface pocket. We also, just to make a long story short, we also analyzed the possible effect of amidine on the spike protein AC2 complex, so considering that amidine can bind also after the complex formation. Complex, I mean spike protein and the AC2 complex. And also in this case, we saw a disrupting effect of amidine, because basically amidine was able to disrupt, to alter, to change, and to reduce the complex stability. So to wrap up this part, this first part of the presentation, our work basically provided a likely mechanism to explain the activity of amidine in preventing the interaction between the spike protein of the old coronavirus and the human cognate receptor AC2. But the $1 billion question is, may it work also on SARS-CoV-2, the new coronavirus? Thomas already said that is extremely important, the analysis of repurposing of pharmacologically active compounds. And actually, there are some evidences in the literature pointing to the possible relevance of amidine also against the new coronavirus. We analyzed the sequence and the conformation and the structure of the spike protein of the old coronavirus and the one of the new coronavirus. And it was not surprising to see how much they are similar. They are similar in terms of primary sequence. They are similar in terms of 3D organization. Also in terms of concerning the surface pocket, the surface cleft we identified. We applied the same approach we used to study the spike protein of the old coronavirus. And also in the case of the new coronavirus, we saw the same contact hotspots, and we saw the same possible binding groove at the protein-protein contact surface. Then we also tried to confirm our data, our predicted data. And the best way to do that is to compare, the computational results with the results published and coming from experimental data. And we were quite happy to see that our results in terms of which are the residues mostly involved in the protein-protein interaction were confirmed independently by in vitro experiments. Independently, I mean that even though our paper was published a couple of months after the paper I put in the slide, I just want to show you that actually the very last bit was calculated on the late of April, and this paper was published in May. So just to say that those methods can be really effective to support and to drive somehow the experimental investigation of COVID. So as I said before, we applied almost the same approach also on the new complex. I mean the complex between the new spike protein and the human target receptor AC2. And we saw that also in this case, amidine was able to bind the surface groove, was able to persist at the spike protein surface. And when we compare the effect of amidine on the protein organization, the effect amidine had on the old spike protein and the new spike protein, we saw that actually amidine could be able to disrupt to a higher extent the spike protein of the new coronavirus compared to the spike protein of the old coronavirus. So in conclusion, just moving to the conclusion, what basically amidine for sure won't be the solution for COVID, but the main outcome of our research was that actually we described a likely mechanism of action to explain the biological activity so for amidine on COVID. And in addition to that, we provided a likely mechanism of action that is worth to be analyzed further to prevent the interaction of COVID with cells. Can this approach be applied also to the new COVID? Of course, yes, they can, they should, and for sure they are applied, and they serve to help the discovery of new active molecules, but also to better understand the mechanisms of action of biologically active molecule, because you have a clear, let's say, view from a mechanistical point of view. And just to say, to give you some additional hints on this specific aspect, and could this approach really work? Of course, yes. Thomas already said something about vaccines for sure are important, but also small molecules can be a really powerful tool to fight the new pandemic, because there are many small molecules that have been described as relevant to target the region of the spike protein that is involved in the contact of AC2. And in addition, recent papers published after our work describe that exactly the region we identified is not region to target if you want to actually and effectively prevent the interaction. Just a couple of slides to give you a taste of the structural basis of, let's say, vaccine and how vaccine may work. So why talking about small molecules when basically the whole world is moving toward the use of vaccines? In my opinion, but as I said, and of course I'm not a physician or epidemiologist, it's important to have a double weapon, to have a plan B, basically. And the plan B is super important in the light of mutations. You can see here a graphical representation of an antibody. Here you can see the Fab region, and you can see here a 3D representation of the Fab region in complex with the spike protein of the new coronavirus. This is a crystallographic structure. You can see here the reference. So the UK variant has, among the others, the mutation in position 501. The mutated form has a tyrosine here. If you look at the antibody spike protein complex, a mutation here, the substitution with a tyrosine should not be able to prevent the interaction. And that's why, actually, from a structural point of view, the UK variant shouldn't be a problem in terms of the efficacy of vaccines. But the story is a bit different, for instance, with the Brazilian variant. Among the Brazilian variant has the mutation in position 417. The point is that in the wild type complex, this is a key region in the antibody spike protein interaction, because you can see here that you have a lysine involved in a polar contact. You have a salt bridge here, a contact that brings a lot of energy in the protein-protein contact. In the Brazilian variant, the lysine is mutated and you have here an arginine. In that case, you don't have such an important contact. So in this case, you may have a weaker interaction, and this might imply a lower efficacy of vaccine. That's why we really need to work also on small molecules, and surely in silico methods can be really helpful also for a better understanding on the vaccine limits and perspective. I just want to thank you for your attention, of course, that's all. And I just want to acknowledge people that work with me. And in particular, I want to acknowledge Professoressa Chiara Dall'Asta and Professor Gianni Galaverna from the University of Parma, and Dr. Dorn from the European Food Safety Authority. Thanks again.
Thank you so much, Luca. Very interesting, of course, and many thanks also for the update on this. So, well, we had the two magnificent presentations. So I was wondering if there are, and hoping there are any questions from the audience. You can just send them to me, or you can speak up, whatever you prefer. Anybody? Or was everything clear? Well, maybe I can start with the first question there for Thomas. Well, Thomas, at the end of the presentation, and you know that I think that's very interesting, you talked about artificial intelligence and a potential role in this research field. But maybe for people not so familiar with this, so can you a little bit elaborate on what exactly would be the role of artificial intelligence in this kind of research? So can you give an example of how this can be used in COVID-19 research?
I mean, if you say it's simple, artificial intelligence is making big sense of big data. So the prerequisite for using artificial intelligence is to have a data set which is really big. And I'm not talking about the large Excel sheet. It is really something which is defined by the 3V of volume. So volume which you can only handle with, let's say, a large computer. The variety, so it's different type of information. And it's also the velocity with which this data are generated, for example, by sensor technologies, by online monitoring certain things. So that's typically the type of data it takes. So what can we use it for? We can use it to develop prognosis for a patient. We can follow up trends of an infection. For example, the analysis of which virus variant now might take over with which time is a typical problem we can apply AI to if you have the underlying data. The problem is AI is you need to have the answer in the data. So say if you take a weather forecast, that's tons of data, and you use them to predict the stock market, you will always get an answer because there's patterns you can find. The problem is it's bullshit. It will not help you in real life. It will not help you forward. So the big problem is that people are forgetting that they can only use the data which contain the answer to squeeze an answer out. And the second problem is that it comes with an enormous bias. And the bias of the data will be the bias of what AI finds. So let's take an example. We will typically have first world data on COVID-19, which go into our analysis. And if we squeeze out data and apply them to third world countries, they will simply not fit. So it is a tool of opportunity. It can help to find new targets like Luca just described. It can help to reposition drugs. These are things which have been published, yeah. But these are, so there's tons of opportunity, but use them carefully and see that it makes sense, yeah.
Okay, thank you for the clear answer on that. So any further questions for Thomas? Well, I have a question here from Samantha. So it's a question for Thomas. So do you know if animals are being used for batch testing of either of the vaccines available in the UK? Of
No.
any of the, sorry?
No, I don't. I was trying to find out about this, which type of pyrogen testing currently is being used. I would very much doubt that they did go back to this, but I assume that they are mainly using the bacterial endotoxin test, which is so the Limulus assay. I found this for the Chinese vaccine. They're using the Limulus assay, which I consider highly problematic. Because the Limulus assay is restricted to gram-negative bacteria, and these RNA vaccines are themselves possibly immunostimulatory. As you know, we have toll-like receptors 9 and others to react to virus structures. So it could very well be that there is immunostimulatory activities, and I very much hope that they're using monocyte activation tests. But I spent several hours on the net. I did not find the information which pyrogen test is being used. Otherwise, I would have inserted into my article, which came out just these days.
Okay, thank you, Thomas. So Samantha, I hope this is an answer to your question. So I have a question for Luca. By the way, Barbara, can you please mute yourself because there is a lot of backgrounds. Thank you. So Luca, you talked at the very end of the presentation about the variants, and I think this is very relevant these days. And I really talk about these days because it is really in the news now. So I was wondering, is this also something that you can model? So there is there is a difference in contamination rates between the different variants. So is this something that also would, let's say, result in increased ACE2 binding? And is this something that you could model with your in silico tools?
It could be, yeah, I can model, sure. Because as we calculated the residues involved in the wild type interaction, let's say, and you can identify the residues involved in the same way, you can also estimate the effect of mutations. Yeah, so you can calculate the interaction, the protein-protein interaction, whatever the proteins are. So the spike protein and the cognate receptor or the antibody. But yes, to answer, yes, definitely we can.
Okay, that's interesting to know. So thank you very much. So are there any other questions? I don't see any more questions in the chat box. There is one question about the availability of the presentations. Well, as I mentioned at the start of this webinar, so this is being recorded and this will be made available on the EUROTOX website. So if there are no more questions, then I think we are at the very end, and it's 2 o'clock sharp. So we ended very good in time. So thank you so much. So obviously I would like to very much thank the speakers, for this excellent presentation and in particular for updating, because this is the second webinar. I also want to thank, well, we didn't hear her too much, but she did a lot of work, Elaine. Elaine, affiliated to EUROTOX, so well, she did a lot of the logistics support, so this is indispensable. So thank you so much, Elaine. And of course, I would like to thank the audience for sticking with us and for your attention. And I hope to see you in person one of these days, so maybe on the next EUROTOX conference. Well, with this, I think I can wish you a good remainder of the day. And again, I hope to see you and thank you for attending. Bye, everybody.